Hello, everyone. My name is Hassan Noroda. This is my journal research presentation for Laboratory 342 Biology with Dr. Sima Chokr. And today we're going to be focusing on targeting myostatin as an adjunct treatment for the preservation of cardiometabolic and skeletal muscle function in type 1 diabetes. And this research is going to focus on how muscle health can influence overall glucose control. Below, I have the article, Noonan 2025, International Journal of Molecular Sciences, Oklahoma State University, Department of Physiological Sciences. So, physiological systems, a little bit of background systems, metabolic regulation, food intake, water balance, endocrine system, skeletal muscle physiology, and glucose homeostasis. Type 1 diabetes, some background, occurs when insulin producing beta cells are destroyed, which leads to high blood sugar, muscle loss, and metabolic imbalance. Since skeletal muscle is the main tissue responsible for glucose uptake, losing muscle mass actually makes blood sugar regulation even worse, makes it way worse. Um, Myostatin is a protein that limits muscle growth, and it's elevated in T1D, which I'll tie back into what about T1D is, as we see on the presentation, which contributes to muscle wasting. A little bit about T1D in type 1 diabetes mellitus cells that produce insulin, known as beta cells, are destroyed. The result of this hyperglycemia, muscle dystrophy, endothelial dysfunction, and metabolic dysfunction. Since muscles are the organs with the highest use of glucose in the body, muscle loss increase difficulty in glucose regulation. So, which ties to our research question and hypothesis. So, the main question is whether deleting or inhibiting myostatin can protect against the negative effects of type 1 diabetes. Does myostatin deletion offer insulin independent protection against the metabolic, skeletal, muscular, and vascular dysfunctions linked in type 1 diabetes? And our hypothesis for today is targeting myostatin via genetic deletion will protect against vascular and renal failure, maintain skeletal muscle function, and stop glucose malfunction in T1D animal. Um, preserve muscle function and protect against other complications even without restoring insulin. So now let's move on to our methods. So our methods, to test this research, researchers used both normal mice and myostatin knockout mice. They induced type 1 diabetes using a chemical and monitored the mice for 28 days. So it was monitored for 28 days post-induction. We had the four groups listed on the slides. They measured blood glucose, HbA1c, muscle strength, vascular function. So vascular function was also measured and metabolic outputs like food and water intake. Um, Muscle function measured via in vivo plantar flexion, and yeah, this was the methods, and I hope you enjoyed the GIF of the laboratory mice, the universal, well-known mice used in science methods and hypothesis, unfortunately. So, some of our results are glucose control. As you see here, A, random feeding glucose, B, plasma insulin, and C, fasting glucose. Just take a look, let that data information figure ooze in. I'll give you a couple of seconds. Awesome. So the results showed that diabetic mice with normal myostatin developed severe hyperglycemia, while the myostatin knockout mice maintained much lower blood glucose levels. So much lower blood glucose levels. Even through both diabetic groups had little to no insulin. Weird. The knockout mice had better glucose tolerance and lower HbA1c, suggesting improved long-term glucose control. So T1D myosin KO mice maintain significantly lower glucose levels. T1D WT mice develop severe hyperglycemia, as mentioned. Over time, insulin is undetectable in both diabetic groups. Physiological, meaning myosin deletion preserves glucose control, independent of insulin. Increased skeletal muscle mass improves whole body glucose uptake. Data shown as the mean plus or minus 7, and p-value is less than or greater than 0.05. So interesting data. Now let's move on to the results number two, muscle function. I'll give you a second to read that and kind of understand the data we have here with the max twitch force, the B max, max tetanic force, and the force frequency, and fatigability. When looking at muscle function, the diabetic mice with myostatin showed significant weakness and reduced force production. In contrast, the myostatin knockout mice preserved muscle strength and performed similarly to non-diabetic controls. This suggests that removing myosin protects against diabetes-related muscle loss. So stimulation frequencies in T1D, but maintained force in T1D, myo-KO failability, fatigability is similar across groups. Significance that endurance is not primarily affected. So skeletal muscle function maintained in T1D. So our discussion in type 1 diabetes, myostatin deletion improves glucose tolerance and helps prevent glycer hyperglycemia, hyperglycemia, glycemia. Sorry. These findings highlight that skeletal muscle is not just a passive target. It's not just a passive target of insulin, but an active regulator of glucose levels. By maintaining muscle mass and function, the body is able to take up more glucose, improving overall metabolic control even without insulin. And preserving muscle function reduces secondary complications of diabetes, closely linked to endocrine controls of glucose, insulin, including insulin signaling and hormone deficiency, demonstrates how this function in the endocrine system can impact both cardiovascular and muscle systems. So, future directions and critical analysis, opinion based, also with some of my own commentary and thoughts. The study used only male mice, which limits how well the findings apply to females. However, there are some limitations. While just mentioned, the study only used male mice, and genetic deletion of myosin isn't exactly how treatment would work on humans. In humans, future studies should test drug-based inhibitors, including female subjects, and long-term and look at long-term effects and additional tissues. Investigate whether combining myosin inhibition with resistance training enhances benefits, assesses long-term effects on cardiovascular and kidney health, study glucose transport mechanism in different muscle fiber types, and definitely explore inhibiting myostatin can reduce development of insulin resistance in type 1 diabetes. In conclusion, targeting myostatin shows strong potential as an additional therapy for type 1 diabetes by preserving muscle and improving glucose regulation. Thank you for your time. I have below the reference for those interested. Thank you so much for listening, and I hope we can take this information and dissect it for everyone's own opinion and study. Thank you.